Viral hemorrhagic septicemia is a virus that is found in the ocean and is killing about 50 species of freshwater and marine fish in the northern hemisphere. To stop this virus, we can take a harmless protein gene from the virus and put it into the DNA of the fish just like a vaccine. Once the fish have the protein gene, they could be resistant to the virus and it can save the fish. The virus affects 50 different species in the ocean, so it can save a lot of fish. The virus can easily be transmitted through water, so it can easily affect the fish. Fish that live in groups can spread the virus quickly. Though saving the fish would create an overpopulation of fish, this could be bad for the ocean, but good for humans because there will be more fish to eat. The virus was around Europe for many years as a carrier. Didn't affect anything. Its early name was Ictvid disease. Description of rainbow troutfish. One of the species of fish this virus affects is the rainbow troutfish. All it will have is a regular looking rainbow trout, but inside its DNA it will have a harmless protein from the virus that will save the fish from the virus if it is attacked by it. Viral hemorrhagic septicemia causes a lot of bad symptoms in fish. External signs are that fish take on darker coloration, the gills may become more pale, and pop-eye. Internal signs are liver may become more pale with hyperemic areas. Hyperemic areas are areas with excessive blood. Kidneys become more red and swollen, spleen is enlarged, and digestive tract may not have any food in it. This picture is an example of a hyperemic area where you can see there's a lot of blood there where it's not supposed to be there. The DNA of viral hemorrhagic septicemia. According to many scientists, the glycoprotein G was the most effective protein to help immunize the rainbow trout from VHS. The peripheral blood leukocytes isolated from the trout immunize against the G protein and kill the VHS infected part. Biotechnology. Scientists will use a gene gun to insert the protein from the virus to the DNA of the rainbow trout. This is how a gene gun works. It takes genes from one organism and shoots it into the other organism, just like a gun shoots bullets. Protein synthesis of viral hemorrhagic septicemia. Once the protein gene is inserted into the DNA of the rainbow trout by a gene gun, it first transfers the protein to form mRNA. Then it will translate the trait or gene that make the fish immune to the virus and carry that protein to the ribosome. The tRNA will transfer amino acids to the ribosome. The mRNA contains the codons and the tRNA contains the anticodons. These two match up with each other to make the proteins. But because of the new trait that the fish will be immune to the virus, it creates a different protein and changes the DNA. This mutation doesn't change the fish entirely, but just makes it immune to the virus. Meiosis of rainbow troutfish. Rainbow troutfish have 58 chromosomes in each cell. When the fish go through reproduction and meiosis, the genetic diversity increases because of meiosis. In the troutfish, it increases even more than in human because we have 46 chromosomes, but the trout have 58. These offspring are very likely to get genes from previous generations. Genetics of rainbow troutfish with viral hemorrhagic septicemia. The recessive trait is immunity of the virus since it's still affecting a lot of fish. As you can see from the Punnett square, the big S represents the virus and the small s represents the immunity. As you can see that the big S is dominant and that the offspring have a 100% chance that they are going to get the virus. All in all, fish that are infected by viral hemorrhagic septicemia can be saved and will be. Thanks for watching.